Hi everyone, today we are going to learn about the urea cycle. Urea cycle is also called Krebs-Henschel cycle, ornithine cycle. Krebs and Henschel after the name of the scientists, and ornithine why? Because it is the first member of the reaction. Now, what is the site of occurrence? Urea cycle takes place in mitochondria and the cytosol. Before starting with it, I'd like to say you that my diagram is a bit clumsy in the right side. All the enzymes which is highlighted with this pink color, these are the mitochondria. These are present in mitochondria and the rest enzymes are present in cytosol. So ये बात याद रखना कि जो हमारी जो enzymes को हम pink से highlight किए हैं वो सारे माइटोकॉन्ड्रिया में है, ठीक है, और अदर जो है नॉन हाईलाइटेड वाला एंजाइम वो सारे हमारे साइटोसोल पे है. नाउ फर्स्ट ऑफ ऑल व्हाट इज दिस यूज और व्हाट इज द सिग्निफिकेंस ऑफ दिस यूरिया साइकिल. हम यूरिया साइकिल क्यों हो रहा है? द वेरी फर्स्ट थिंग इज दैट अमोनिया. सी हियर, अमोनिया और द फ्री अमोनिया इज वेरी 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 मच डेंजरस फॉर आवर बॉडी. इट इज वेरी मच टॉक्सिक. हेंस to remove this free ammonia, body is undergoing this urea cycle. Now let us first come to the cycle itself. Ammonia plus carbon dioxide plus water, it's giving carbamoyl phosphate. In this step 1, 2 ATP are used. What is the enzyme? It is carbamoyl phosphate synthetase 1. Why 1? Because there is another also carbamoyl phosphate synthetase 2 which is involved in pyrimidine nucleotide synthesis. So if you say only carbamoyl phosphate synthetase, you will be not so much appropriate. You have to say carbamoyl phosphate synthetase 1. And because here in this reaction ATP is used, so we are using the term synthetase. Okay? Otherwise it would be synthase. But here ATP is used, so it is going to be synthetase. And this is the rate limiting enzyme of the entire urea cycle. Now carbamoyl phosphate, it is changed with ornithine. Carbamoyl phosphate with ornithine, it is giving us citrulline, right? Using the enzyme ornithine transcarbamylase. Citrulline now with the help of the citrulline transporter, it is moving out of the mitochondria. Then citrulline along with the aspartate, it is forming arginosuccinate using the enzyme arginosuccinate synthetase. So that means again ATP will be used here. So here 1 ATP is changed into 1 AMP, which is equivalent to 2 ATP being used in the reaction. Okay, we will see this part in the energetics. Now aspartate. Aspartate is coming from where? It is coming from somewhere of the TCA cycle. So this urea cycle, and in the side you are seeing one cycle that is a part of the TCA cycle. These are interlinked. It is often called the urea bicycle for this purpose. Okay. Now coming to arginosuccinate. Arginosuccinate with the enzyme arginosuccinate lyase, it is getting converted into arginine. And in this reaction also fumarate is getting released. So this fumarate, with this help of the fumarase, it is converting into malate. Malate, in the help of malate dehydrogenase, it is converting into oxaloacetate. And oxaloacetate, it is converted into the aspartate using the aspartate aminotransferase. Then again this aspartate aminotransferase, it again enters along with the citrulline to produce the arginosuccinate. Now very important here, all the highlighted enzymes, this fumarase, malate dehydrogenase, these are present in the mitochondria, whereas the non-highlighted enzymes like the aspartate aminotransferase, then in this case, and all these are as arginosuccinate lyase, arginosuccinate synthesis, every non-highlighted one is present in the cytosol. Now, so yes, So now arginosuccinate, it is converted into arginine. Now arginine, it is converted into ornithine, and in this process urea is released. Okay, this is hydrolysis and urea is released with the help of the enzyme arginase. Ornithine, with the help of ornithine transporter, it again moves back into the mitochondria. Very important one you will notice in the entire cycle, that ornithine is the first member of the cycle, and again ornithine is regenerated. So no part of ornithine is incorporated or it is used. Okay, that is, ornithine can be called a catalyst because it enters the reaction and it is regenerated here. So what can we call this? Entire urea cycle can be said to be that is ammonia with carbon dioxide with aspartate. It is giving us fumarate and urea. This is actually our overall reaction of the urea cycle. Now let us come to the energetics part. Energetics part C. Step 1, the source is ATP. ATP gained is -2. What does it imply? It implies that 2 ATP is utilized. So where is the step 1? Let us see where is the step 1. Yes, here's in step 1 2 ATP is used. So ATP gained is -2. Next, in step 3, ATP is converted to AMP, which is again equivalent to 2 ATP used. That is this one, this step. Now coming to the step 7, NADH. So NADH, when it enters the electron transport chain, it will give us 2.5 ATP. So it is net is -4 plus 2.5, it gives -1.5. So it is equivalent to ATP used 1.5 ATP, that is the 1.5 high energy phosphate bonds. So this is actually our energetics of the urea cycle. Now coming to the regulation of the urea cycle. Regulation of the urea cycle, it is that when there is starvation, then what happens in prolonged fasting, prolonged starvation, our muscle protein starts breaking down, that is muscle proteolysis happens. When there is muscle proteolysis, then alanine of this muscle, it is converted into glucose by the liberation of ammonia. And I have told you that free ammonia is very much dangerous and toxic for the body. So this ammonia enters the urea cycle. Urea is much more harmless than ammonia. Okay, then it enters the urea cycle and this rate of the urea cycle increases. Again, if there is high intake of the dietary proteins, it also results in high or the more amount of urea cycle. Again, these are the coarse regulations. Okay, there is also fine regulation working in this entire cycle, which is the N-acetyl glutamate. N-acetyl glutamate, by stimulating this carbamyl phosphate synthetase, by having a positive impact on this carbamyl phosphate synthetase 1, it will increase the urea cycle. Okay, so this is all about the urea cycle. I hope you have mastered it. Stay tuned with this channel. If you like the video, please click on the like button, click on the subscribe button, and share with as many of your friends, your sisters, brothers, whoever you like, if you think that it's worth sharing. Stay tuned for more such videos and keep on suggesting what I should come up with in the comment section. Thank you.